Hi, I'm Daniel Kim. I'm a student from OVC Class of 2017, and this summer I worked at Department of Pathobiology under supervision of Dr. Darren Wood. My research involved this protein called thrombomodulin, and the work I'm presenting here today is a retrospective evaluation of thrombomodulin expression in tissues from dogs with systemic inflammatory diseases. Thrombomodulin is a membrane-bound glycoprotein which is primarily expressed on endothelial cells, and it has been very well known for its function in protein C anticoagulation pathway. And this diagram is basically showing you a coagulation pathway starting with the platelet aggregation leading to activation of thrombin and activation of fibrin clot formation. And what thrombomodulin does is that it will bind to thrombin and activate the protein called protein C, which will inhibit the various clotting factors to stop this coagulation pathway. What's interesting about this protein is that in human medicine, especially in the patients with severe inflammatory diseases, thrombomodulin has shown to be downregulated and is released into circulation, increasing the risk of thrombosis as well as disseminated intravascular coagulation. This protein is also expressed in dogs. However, there has been very minimal studies done to look at the thrombomodulin expression in canine tissues, and also the function of thrombomodulin during the inflammation has not been investigated. So in this study, we wanted to evaluate endothelial thrombomodulin expression in tissues from dogs with or without systemic inflammatory diseases. So basically what we did was we collected tissue samples of lung, spleen, and liver through postmortem of normal dogs or dogs with systemic inflammatory diseases, and these tissue samples were evaluated for thrombomodulin expression using immunohistochemistry. For normal dogs, the tissue samples were collected from animal health laboratory that showed no significant lesions of diseases, whereas for dogs with systemic inflammatory diseases, we searched hospital records from Ontario Veterinary College Health Sciences Center years in between 2006 to 2011 to obtain tissue samples from dogs that were diagnosed with sepsis or systemic inflammatory response syndrome due to pancreatitis. Basically, tissue samples we collected were from formalin-fixed, paraffin-embedded tissue blocks. So with those tissue blocks, we created the tissue sections and then immunostained them with thrombomodulin antibody for thrombomodulin expression evaluation. And thrombomodulin expression was evaluated using a semi-quantitative method, which is a scoring method that I have came up with based on the previous literature with some modifications. So basically the idea was look at the thrombomodulin staining on these tissue sections and score them from 0 to 3, 0 being negative, 1 being weak, 2 being moderate, and 3 being the strong for 6 randomly selected fields of view. This task was performed by myself as well as Dr. Darren Wood independently. However, for liver, in our preliminary study, we found that tissue sections from livers in overall do not stain very well with thrombomodulin, which was actually consistent with the previous findings in human as well as in mice. So we took different approach. That is, the total number of vessels present in each section was counted, and the number of thrombomodulin positive vessels was used to calculate the percentage vessels staining with thrombomodulin. And here are my results. This column is basically showing you a mean score that were produced from thrombomodulin staining for lung and spleen. And the third graph here is showing you a percentage vessels that were stained with thrombomodulin in liver sections. And the column over here is basically the representative IHC images that corresponds to these graphs. So I will slowly walk you through my results, starting with the lung. tissue sections from normal dogs showed strong thrombomodulin staining with the mean score of 2.64. And as you can see from its corresponding image, you can see strong thrombomodulin staining throughout the endothelial cells lining alveoli as well as pulmonary vessels. The staining pattern was also very continuous and homogeneous. On the other hand, the tissues from dogs with sepsis or systemic inflammatory response syndrome, they had a mean score of 1.28 and 0.91. And as you can see from their corresponding images, thrombomodulin staining was significantly reduced on both sepsis and systemic inflammatory response syndrome dogs. And the staining pattern was very discontinuous and not homogeneous. Similarly, in spleen, tissue sections from normal dogs showed strong thrombomodulin staining with the mean score of 2.41. And as you can see from its corresponding image, almost always the thrombomodulin staining was positive on endothelial of most blood vessels and stroma within the spleen. However, in the dogs for sepsis and systemic inflammatory response syndrome, they had a mean score of 1.18 and 0.96. And as you can see from their corresponding images, thrombomodulin staining was significantly reduced in both of the cases. And the central arterioles did not stain, and also the white pulp showed the little to none thrombomodulin staining. And also, thrombomodulin staining patterns were more diffuse and discontinuous throughout the sections. And lastly, the liver. As I mentioned earlier, all of the liver tissue sections had poor thrombomodulin staining. And even one of the very well-stained even one of the most well-stained vessels will have huge variation in the score from 1 to 3, and it will have appearance something like this. However, though, in normal dogs, there was more than 50% of thrombomodulin staining in the liver sections, whereas in the cases of sepsis or systemic inflammatory response syndrome, they only had 17.6 and 13.5% thrombomodulin staining. And basically, if you have the tissue section stained with thrombomodulin from these dogs, what you will see is lack of thrombomodulin staining, as you can see from these images across the tissue sections. So basically, our data show that thrombomodulin expression is significantly reduced in lung, spleen, and liver in dogs with sepsis or systemic inflammatory response syndrome. This may be due to downregulation of thrombomodulin expression on endothelial cells in response to cytokines produced by migrating inflammatory cells, or direct cleavage of extracellular domain of thrombomodulin by neutrophil-derived proteases and oxygen radicals. So this was a very interesting study because now we can do a case-controlled studies to look for serum thrombomodulin as a prognostic or diagnostic tool for dogs with systemic inflammatory diseases. So that sums up my research project. And I'd like to thank William Sears for helping me with statistics, and Susan from AHL for performing IHC, and of course my funding, Petrus Fund, for supporting this project. Thank you.